Okay, so today we're going to talk about DNA structure and replication. So make sure that you're drawing lots of diagrams as you're watching these notes and that you're jotting down any questions that you may have that we can go over in class. So let's talk about DNA structure first. We've already done DNA structure. We did it back with organic molecules. We talked about the structure of DNA, but I just want to review it and make sure that we remember everything. So remember that they're made up of nucleotides. And a nucleotide consists of three things: a deoxyribose sugar, a phosphate, and then four bases. And the bases again are adenine, cytosine, guanine, and thymine. Okay, so hopefully that's a review, and now that we've talked about it again, that's coming back to you. So here's the structure of the nucleotide again. So remember that you have the phosphate group attached to the sugar, which is attached to the base. These are purines. Remember that a purine means that the base has two rings. So adenine and guanine are your purines. The pyrimidines have only one base, and so that's cytosine and thymine. Remember when they bond together, adenine bonds with thymine. Remember the word at helps you remember that. And guanine pairs with cytosine. So here's our structure again. So we have the outside, remember it's considered a double helix. So we have the outside ring being the phosphate, the sugar, and a base. That's one nucleotide in the strand of DNA. So cytosine connects with guanine, and that there are three hydrogen bonds between those two strands connecting them. Our next nucleotide, adenine, pairs with thymine, and there's only two hydrogen bonds connecting them. So three between cytosine and guanine, and two between adenine and thymine. Okay, so now let's talk about what all that has to do with DNA replication. So with DNA replication, the genetic information is duplicated, and that has to be completed before cell division can take place. And that's what we're focusing on with this unit, is how we get new cells. So in order to get new cells, we have to replicate our DNA first. So it's going to occur during what's called the S phase of the cell cycle, which we'll talk a lot more about later. But just for now, make sure you know it's in the S phase, which stands for synthesis, because we're making DNA. In prokaryotes, remember, our bacteria, it starts at a single point and proceeds until the chromosome is replicated. Remember we said one of the differences is that the DNA is much smaller, it's circular, so it can just start at one point and keep going and it's finished. Where eukaryotes, like our cells, it starts at many, many different places along the DNA chain, and it can go in both directions. And that's because the DNA is so much bigger, and in order to be able to get it done in a timely manner, we need to do it that way. Okay, so let's talk about the structure. So this is a nucleotide. We have our phosphate, our sugar, and our base. These numbers are really important, so make sure that you have this drawn correctly and that you have the numbers labeled. So remember that we said that it's a 5-carbon sugar, and we have to number each carbon. So carbon 1 is where we attach the base. Carbon 2 doesn't have anything attached. So let's just review, and we'll talk about the numbering in a second. So one nucleotide is one phosphate connected here to the 5', one sugar, which is where all the numbers are, and then one base, which is attaches to the 1'. So let's look at a strand of DNA in our numbering. The numbers that we're concerned with are the 1, the 3, and the 5. The 2 and the 4 don't connect to anything other than within the sugar, so we're not really going to focus on those. The 1, the 3, and the 5 are what's important. So it's always the same. The nitrogen base attaches to the 1, and the phosphate is at the 5 of one nucleotide. So at the 1 and the 5 is one nucleotide. The 3 is also important because it attaches to the phosphate of the next nucleotide. So that's also important. So 1 and 5 are within the same nucleotide, and then 3 is involved with bonding to the next nucleotide. So when we read DNA, it's always read from a 5' to a 3' direction. Okay, always from 5' to 3'. So let's look at it. Remember that the bases are complementary. So the top strand runs 5' to 3' from left to right, and the bottom strand runs 5' to 3' from right to left because they're upside down, so it runs in opposite directions. So once you have your model completed, you'll see that they're running upside down from each other. So A bonds with T, C bonds with G. And if you take out all of the phosphates and the sugars and everything to make it simpler, you know that 5 to 3 is strand 1, and then 5 to 3 strand 2, just going in the opposite direction. And that's how we read it. Okay, so let's talk about the steps involved with DNA replication now that we know that how everything is arranged. So we have our 2 strands of DNA. And remember, and it's really one of the unique things about DNA is that if we have one strand, it determines what the second strand is, so it can replicate itself. So the first thing that has to happen is that all of these hydrogen bonds have to be broken. And that occurs by the enzyme DNA helicase. So DNA helicase unzips the DNA is usually how we say it. It unzips your genes. So it's a kind of a clever way to help you remember that DNA helicase unzips the genes. So now you have 2 separate strands, right? They're no longer connected together. And so then those are called templates because you can tell what bases should be going in here or what nucleotides. So if you have a thymine nucleotide, you know connected to it would be adenine. And then on this one, you have an adenine nucleotide, so thymine will be connected to it. Okay, so that's the first step. The second step is that we will add in the correct bases, and that's using the enzyme DNA polymerase. So that's going to add in the correct nucleotide bases going in the 5' to 3' direction is how they get added in, 5' to 3'. Okay? So it adds in the bases 5' to 3'. And then proofread. You have to proofread, make sure that there aren't any mistakes. DNA polymerase will then go back and check to make sure that they did it correctly. So if something doesn't go correctly, then they'll take it out and they will replace it with a correct nucleotide. So our final product, which put all together, see, so it's so easy. 3 steps. You unzip the genes, you add in the correct nucleotides, and you proofread it to make sure you didn't make any mistakes. So when replication is complete, we end up with 2 identical strands of DNA. Each molecule contains 1 original strand and 1 new strand. And that's called semi-conservative replication. So here's what it looks like as it's being added in. Here's our new strand being added to our original strand. Here's our original strand with the new strand being added in. Okay? So 2 new strands. So now we can make new cells. And that's our whole goal, is to be able to reproduce and get more cells. So let me show you this little animation to help make sure that you really understand what's going on.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it.
So remember, that's
DNA polymerase
the first
3
step.
is the major enzyme involved in DNA replication. DNA polymerase.
Okay, so the first step, again, remember, was that we unzipped the genes. DNA helicase came in and did that. Now DNA polymerase is going to be adding in the correct nucleotides.
Polymerase 3 can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides.
This is a little more information than you need to know, but you can still see what's happening.
DNA polymerase 3 can then add deoxyribonucleotides
to
synthesize the new complementary strand of DNA.
So you're adding in a new strand to the original strand.
Because the 2 parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments.
Again, this part's a little bit more information than you need to know right now.
RNA primer on the lagging strand, it is replaced by
It's still good
DNA
information.
polymerase 1, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment.
Remember, on the other side, it's being added in continuously.
For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Okay, so what I want you to remember is that DNA replication is really simple. We have 3 steps. We have our DNA separating, and remember that's DNA helicase that's going to separate them. We have DNA polymerase come in and add the correct base, and then we proofread to make sure that that's what's occurring, or to make sure that we added in the correct bases. So adenine matches up with thymine, and cytosine matches up with guanine. And that's all. So write down any questions that you have. Make sure that you have your drawings completed and your numbers labeled so that you know which end's the 5' and the 3'. And we'll talk about this in class.